And I think my talk's probably going to be a bit more, I guess, practical rather than scientific. And it's just a way of sharing some experiences we had trying to establish live food colonies for mountain chicken frogs in a conservation breeding program that was established on the island of Dominica. So just a little introduction to the species. So the mountain chicken frog is the largest native frog in the Caribbean. They can weigh over 1 kilogram, and they were an important source of meat for local people. They're a culturally significant species, especially on the island of Dominica, where they feature on the nation's coat of arms. They're assessed as critically endangered, and previously they were found on up to 7 different Caribbean islands, but they're now currently restricted to the small island of Montserrat and Dominica. So the species was historically threatened by overexploitation for meat and also habitat loss. And on the island of Montserrat in 1995, a volcanic eruption threatened the species and its very existence. So in response to this, a conservation breeding program was established in Europe at the request of the Montserratian authorities. And a huge amount of what we know about the species is due to the early attempts to rear and breed them in Jersey Zoo in the Channel Islands. So a lot of what we know about the species comes from there. But unfortunately in 2000 on the island, or the early 2000s on Dominica, chytrid arrived. So as you probably all know, the pathogen, in this case BD, which causes the disease amphibian chytridiomycosis, arrived on Dominica in the early 2000s, and then later it reached Montserrat in 2008. And as a result, this frog underwent precipitous population declines. Over 85% of populations were lost within 18 months. So this was the fastest ever documented decline of a vertebrate, we believe. And especially on the island of Dominica, the range massively contracted. So you can see the light, the light green area. Sorry, the grey and the light green area shows the former distribution of the species, but they're just now, it's restricted to these kind of scattered dark green polygons you can just about make out on the map. And on the island of Montserrat, the species is functionally extinct or potentially extinct in the wild. So this led to the formation of the Mountain Chicken Recovery Programme, which consists of national governments, zoos, academic institutions. And together we've developed and are in the process of implementing an action plan that will drive, or hopefully drive, the recovery of the species. And this is also guided, the captive ex situ program in Europe is also managed by a stud book, which is coordinated by Gerardo Garcia in Chester Zoo. And in response to the precipitous declines on Dominica, it was also considered prudent to establish a conservation breeding facility on the island. And this was developed with the collaboration and lead of the forestry department there, and with the assistance of several European zoos. And in tandem, and when BD arrived on the island of Montserrat, another population was established in European zoos as a safety net. But we are also guided by conservation needs assessment. So the amphibian arc conservation needs assessment recommends this species for rescue, as well as in situ conservation, in situ research, conservation education, and biobanking. But we strongly believe that the best place for the conservation breeding facility would be in the range state, which is in line with, I guess, the philosophy of amphibian arc, as well as the amphibian conservation action plan. And this species is also an evolutionary distinct and globally endangered species, or an edge species, so a global priority for conservation. So that's just a bit of a background to kind of why we started this project. So the mountain chicken frog, as I mentioned, is a really large frog. can consume more than 100 different invertebrates a week. And so the historic population on Dominica of 100,000 individual frogs could have consumed over 10 million invertebrates in a single week. So what you can guess from this is they had a very important role in the ecosystem. But when it comes to managing a species such as this in captivity, this proves to be quite a challenge. But fortunately for us, this species has a very specialized reproductive mode, and we don't have to really worry about feeding the tadpoles. And this is why. So for those of you that are not familiar with this species, this is a mountain chicken frog. You can make out her cloaca here with the tadpoles swarming all over it, and she's feeding the tadpoles infertile eggs. So this species breed in underground nest chambers where they produce a foam nest. The tadpoles develop within the nest, and they're entirely fed by these infertile eggs. So the mother kind of feeds them once or twice a week for a period of about a month until they metamorphose. And I think this also emphasizes, I guess, the value of maintaining amphibians in an ex situ setting, because these behaviors would be very, very difficult to observe in the wild. And just to emphasize, a lot of what we know about the species comes from these observations in a zoo setting. So before the mountain chicken was a critically endangered species, I mentioned it was incredibly abundant, and a study by Brooks looked at the stomach contents of nearly 400 individual frogs on the island of Dominica. So these frogs were killed and their stomach contents were analysed, and it shows a huge diversity of different prey items in the diet of the mountain chicken. And this therefore means it's quite complicated to kind of replicate these diets in a captive environment. Really, really challenging. So from small to relatively small invertebrates right up to small vertebrates. So this is something that we really need to consider when maintaining the species in a conservation breeding setting. So when the conservation breeding facility was built and designed on the island of Dominica, we had to choose local species of invertebrates for a number of different factors. One, as was kind of mentioned in the previous talk, we didn't want to kind of send invertebrates that are widely cultured, say, for example, in zoos in Europe, to the Caribbean, because these could become pest species, and that could be economically damaging to the islands. It might be quite challenging for those invertebrates we would want the invertebrates to be able to easily acclimatize to local environmental conditions. It would also eliminate purchasing and importation costs, which to a small island in the Caribbean might be quite considerable, as well as the facility being able to operate and the frogs to be fed in case of things like tropical storms affecting importation or other unforeseen circumstances. And if we were to use local species of invertebrate for live food and culture these invertebrates, it would be relatively simple and cost effective to restock any cultures if they became depleted. We also had some additional considerations. We wanted to select species where we perceived that they might be able to rapidly reproduce. We wanted to give preference for the species that have been documented to form part of the natural diet following the study I just referred to. We wanted large-sized invertebrates because the mountain chicken itself is a large frog, so they do tend to feed on large prey items. We wanted to try and avoid invertebrates that were territorial, so we could raise them at high densities. And we wanted to avoid invertebrates that might pose a risk to the mountain chicken frogs, so things that might, feed on the frogs. And also the final consideration was the time needed to maintain and take care of these live food colonies. So that's quite a steep wish list, I guess, but we were very optimistic and perhaps a little bit naive at the start of this. So this is the conservation breeding facility that was developed, and it was constructed in 2007, And you can see in the bottom left, there's the map of the facility, and the area highlighted in red is the area that was dedicated to the production, solely for the production of the live food. So probably about 25% of the space was dedicated for this purpose. So that was quite a considerable, I guess, investment in terms of the design of the facility. And so then we had to kind of set about sourcing the invertebrates that we would use. And there's I didn't really have many photos of this. There's a photo of me collecting some invertebrates there, but I have to say most of the work was done prior to my brief time on Dominica, and was done by other people. So I just want to acknowledge the huge number of people that have been involved in this. But some of the invertebrates were collected by hand in local forests at night, and for some species this was relatively easy. There's an endemic Carabacusta cricket and some of the slugs. But the diversity of species, or the number of individuals that we encountered at night in these kind of forest strips, was relatively low, and quite difficult to collect sufficient numbers to really establish viable live food colonies. And I just want to add something here, is that quite often we didn't know which species we were dealing with, especially with things when we get on later to things like the millipedes that we attempted to culture. We've got no idea what they actually were. So it was a bit of a step into the unknown. We also attempted to trap invertebrates, especially in agricultural land, and we were able to actually detect one species, Gryllus assimilis, which is actually a widely cultured live food species throughout the world. But our colonies were actually founded from a single pair. Despite weeks and weeks of searching, we only ever found single male and a single female, which was quite lucky. But we also wanted to consider the fact that there might be pesticides used in these agricultural landscapes, and so we didn't want to repeatedly kind of collect individuals from this, because we didn't want to transfer animals that had been in this landscape to the facility where they'd be consumed by the frogs, and there'd be a risk of contamination. Another consideration we kind of thought about early on was the use of light traps, or even having light traps around the facility for the frogs. But bear in mind, as the primary threat to this species is the chytrid fungus, and the frogs developing chytridiomycosis, we had to make the enclosures, I guess, we didn't want animals to be able to readily enter the enclosures, including other frogs that might be reservoirs of infection. So we did attempt to use lighting to attract insects just purely for collection, and that might be fine in the field, but actually in the enclosures themselves, they were really, really tall. So most flying insects would be well above would kind of occupy the space well above the frogs, and would probably not be accessible for the frogs to feed on. But we were willing to make exceptions for the flying insects. We located a single banana cockroach just by chance, and we hoped that these would be amenable to culture. But again, we only ever found a single individual, so that wasn't viable. But also actually talking to local people really helped us identify potential sources or potential species we could culture. So a chance meeting led us to someone that maintained chickens on the island, and they had reportedly loads of cockroaches in their chicken coop. And when we went there to inspect them, another quite commonly cultured live food species was encountered, the blabberous cockroach, and we never saw these anywhere else other than this single chicken coop. And it was likely that these cockroaches were imported with the chicken feed that came from Venezuela. And so we were able to establish colonies for about 8 species of invertebrate in this facility on Dominica, but some far, far more successfully than others. So typically it was those species that are already widely cultured, I guess, because they're amenable to that. It's economically viable to culture these in other countries. So things like the Gryllus acimus and the Blavarus, but other species less so. And then I mentioned earlier that vertebrates are also consumed by mountain chicken frogs. But we have, I guess, an institutional policy whereby UK standards of ethics are applied in all of our activities overseas. And in the UK, it's not permitted to feed live vertebrates without veterinary supervision. And even then, only under exceptional circumstances, i.e. that the target species might die without this intervention and provision. And furthermore, some of the reptiles that occur on Dominica are threatened in their own right. So that's something that wasn't very palatable for us and something we decided we would not be willing to do. And the other thing, as I mentioned earlier, was the very small frogs, the Eleutherodactylus, that occur on Dominica. There's a couple of species there, very, very common. These could have potentially, they're known from the diet, they could have potentially been a source of food. But we do know from our wider research on the island that these are really the reservoir of infection. So prevalence at certain sites of BD, about 50% of the frogs were infected. They don't really seem to show any clinical signs of disease. So actually, we went to great efforts to make sure that these frogs couldn't gain access into the facility. So making sure all the drains were covered with mesh. For example, all the doors were tightly fitting, because the last thing we wanted were these frogs to enter the facility. So that was an additional consideration in the establishment of our feeding protocols. So once we'd got some of our cultured invertebrates, we developed just a standard system of rearing live food. So for the crickets, for example, the boxes were organized by Instar, and we rotated the boxes as part of like a breeding line of crickets. But I have to say that the care and the time required to look after the invertebrates far exceeded the time that we took taking care of the frogs. And I have to say, it wasn't the task that everyone found the most enjoyable. People wanted to work with the frogs rather than the invertebrate colonies. So yeah. And then later on, we made some additional refinements to the way we managed these colonies. So at the beginning, we didn't have lights above the cricket enclosures, which we later provisioned. But because this project was, for example, ZSL provided volunteer support. I was a volunteer on this project at the time. We didn't provide support for the actual running costs of the facility. That came from the local government. So adding things like lighting for the live food and stuff was an additional financial burden, and layers of bureaucracy had to be gone through to get these things approved. And I guess there were some restrictions with what we could use for containers and for tubs for rearing these invertebrates. We had to use what was locally available, and there wasn't often a huge amount of choice on a relatively small island. But we found things like these large waste bins to be quite effective for rearing things like the Blavarus cockroaches. And then, as I mentioned, ZSL provided a kind of series of volunteers to help get this project up and running to support our partners in the Caribbean. And so it was really important that we developed standardized ways of caring for our invertebrates, and a live food manual was developed for the facility so that the care of the colonies could be consistent, husbandry drift would be avoided, and we wanted this continuity of care because of this staff and volunteer turnover. However, I'm just now going to focus on some of the challenges, and there were several of them. Some of the key species, so this cricket on the left, this forest cricket, which is one of the most frequently consumed items according to the Brooks study. The mountain chickens is just like a clear staple for them. It proved to be really, really challenging to breed and replenish, probably because they can be quite territorial. So we had to replenish stock on quite a frequent basis, which was really, really time consuming to get enough to feed the frogs in the facility. And the same for we called them velvet slugs. These slugs were difficult to culture, and we had to collect those quite often as well. And we were very mindful of the fact that every time we collected something from the forest, transported it, and these items or animals entered the facility, it represented a potential risk for pathogen pollution. So we tried to avoid it as much as possible, but in many cases, it was absolutely kind of vital that this happened to ensure the frogs were fed. Pest species were a problem, and probably the biggest one were the rats. So rats kind of damaged the infrastructure through gnawing through roofs and through the walls. And they also consumed the food that was meant for the insects. So fighting with rats was a constant battle. And also anolis lizards did enter the facility from time to time, and they would access the live food colonies and just sit there and become gradually stuffed throughout the day. So we had to periodically remove those. Ants were another potential issue, and something we had strategies in mind to prevent, but fortunately not one we actually had to contend with. And then there were also periodic unexplained die-offs of the invertebrates, especially at that kind of new stage from collecting the invertebrate founding stock from nature and trying to culture them. We'd get initial good growth of the colonies, but colonies collapsed and just a few individuals remaining. And then it seems that over time that they would recover and become a bit more robust. And as time went on, they were less prone to collapse. But I don't really know the reason for this. And I'd be very interested to hear anyone's theories. Another challenge was feeding the live food itself. Again, because this facility was government run, so all of the food had to be paid for and budgeted for the project. And rapidly expanding live food colonies obviously need much more food. And then there were some fluctuating availability of some of the key diets that we were able to offer the live food, so things such as fish flake and pellets. That was a challenge as well, because we were not 100% that these items themselves are really that nutritionally sound, but again, the restrictions of working on a relatively small island, we had to use what was locally available to feed the invertebrates. So actually, we couldn't actually offer the live food species. We went to offer them the mountain chickens, the invertebrates that they might consume in the wild, and therefore a natural diet. But it probably wasn't really a natural diet, because the live food itself was not eating its own natural diet. And then we wanted to offer dietary supplements, but again, these were not locally available, and they were also quite difficult to import. And we didn't at the time really have any reference data to know if our diet was nutritionally sound, which led to additional research, which I believe will be covered by another speaker, my colleague Amanda Ferguson, later on in this series. So I won't dwell on that any further. Another kind of, I guess, nutrition and health consideration was that of metabolic bone disease. So nutritional secondary hypothyroidism, a form of MBD, has been documented in captive mountain chickens that have been reared and maintained on diets that have been supplemented with D3, but were not provided with UVB radiation. However, frogs that have been, these are in European facilities, sorry, I should add, frogs that historically exhibited clinical signs of this metabolic bone disease with I guess, skeletal ill health, were able to remineralize their skeletons once they were provided with UVB, as well as a D3 containing dietary supplement. But these malformations are permanent. but frogs that are reared on both with diets containing D3 and UVB provision are skeletally healthy. So I think we can say from studies done in zoos, we demonstrate that the UVB provision is crucial for the health and welfare of captive mountain chicken frogs. So we were very conscious to try and ensure that our frogs were healthy, because ultimately they might be translocated. But it was very difficult to assess skeletal health because we were unable to even radiograph or take blood samples from the frogs on the island. On Dominica, this is an image of one of the pens where the mountain chickens were reared. We were able to provide limited UVB radiation using lamps that had to be brought over in the luggage when people travelled usually from the UK, because again, the UVB emitting lamps were not locally available. And so it's quite difficult to have a dependable supply of these lamps. And natural sunlight did enter through the enclosures. You can see the mesh wall at the back there, but not directly. And so we made the decision to actually try and expose some of the frogs to direct sunlight, but with the option of them to retreat periodically in large enclosures during the day, which we held outside. Again, assessing this husbandry intervention in terms of skeletal health was a real challenge and not possible, but we didn't observe any clinical signs of metabolic bone disease in any of the frogs at any time. And another consideration was the time of day in which we fed the frogs. Obviously, when you feed the frogs, you want the food item to be kind of offered at the optimal time when the frogs are active and when the live food itself is well fed, and if you're using dietary supplements, to be well dusted. But the facility itself was quite isolated in the botanical gardens of the island's capital, and it wasn't logistically possible or even safe for members of staff, especially if they're by themselves, to access the facility to feed the frogs during their peak activity period. So this is a compromise, but safety and security had to come first. But we learned some pretty cool things along the way. So one of the frogs that was imported it was actually seized from a local hunter, and a hunting of the species is now illegal. It was brought into the facility and passed feces which contained the leeward ground snake, part of the leeward ground snake in the fecal matter, and this was a new record, a new dietary item from the mountain chicken, which is quite novel. So I just want to give you a kind of a timeline for these activities. So early 2000s on Dominica, mountain chicken frogs were found dead, and the cause of their death was determined to be chytridiomycosis. And then it wasn't until 2007 that there was sufficient momentum to build the facility and fund the facility. So that's 7 years. And then we needed to establish the live food colonies. And given how much the mountain chickens eat, that was a considerable undertaking to make sure we had viable live food colonies where we felt comfortable that we could, with a clear conscience, bring the first frogs into captivity. Unfortunately, in this time, from about 2006 to 2010, no mountain chickens were actually seen alive on Dominica, and there was a period of time where we feared they may have disappeared from the island. But fortunately, in 2011, frogs were once again sighted, and we brought frogs into the facility for the first time. So I just wanted to highlight here that it's really important not to underestimate the length of time it may take to establish these live food colonies, especially with large numbers of large frogs. So I just want to go through some of the live food items. So some of these key criteria: we wanted native species that were known from the wild diet. We wanted sustainable populations, and obviously a consideration I didn't mention earlier, we wanted the food to be consumed readily by the mountain chicken frog. And you can see actually from the list of species that we were able to culture, not one species really met all of these criteria. And what we actually found is, whilst we didn't need to import exotic species to use as live food, most of the species that were the most suitable for culture were locally collected, but non-native species themselves. So the wild diet could not be fully replicated in captivity. So this is, I guess, a bit of a compromise here. So we managed to feed the frogs; they were healthy, but this benefit of having the wild diet just wasn't achievable. Many of the invertebrates were not amenable to culture, but we didn't have any frogs showing any evidence of nutritional disease over the 7 or so years that this program was running. We also had to liaise with the relevant ministries, because I mentioned many of the species that we were able to culture appeared to be non-native species, and some of them were potentially new arrivals to the island. And so we had to make sure the local ministries were happy with what we were doing, and that we weren't actually facilitating or aiding the establishment of newly arrived and potentially pest invertebrate species. So during the I just wanted to highlight here that it requires a large footprint. During the time that the facility operated on Dominica, we were unfortunately unable to successfully rear any mountain chicken frogs. The greatest number of frogs that we maintained at the facility at any one time was 11 different individual frogs, and we were at full. Live food was bursting out of those rooms, out of that 25% footprint that I've shown on that figure. And should we have been in the fortunate situation to have bred the frogs, we would have needed a much larger footprint to actually have viable colonies that would support the number of frogs needed for a full-blown translocation, if we were in the fortunate situation to be there. So I just want to briefly explain some of the reasons I think maybe we were unsuccessful in breeding the frogs, and that was because whilst it's very laudable to establish conservation breeding facilities in range states, one of the risks in doing so is the very close proximity to the threat process itself. And in this case, it's BD and the chytrid fungus. And there were unfortunately periodic outbreaks of chytridiomycosis, which we managed to treat successfully. But these often coincided with the start of the breeding season, and the disease events likely hindered the breeding. This example with regard to the footprint required for the live food colonies is not unique. So this other figure is of the facility in Matinju in Madagascar, and they've also had to dedicate a similar amount of space to the production of live food. So I now want to explain what happened to the facility, and unfortunately Dominica was really badly hit by Hurricane Maria in 2017, and huge tracts of forests, you can see before and after photos in the bottom right, were really badly damaged, and the conservation breeding facility was almost irreparably damaged. It's currently unclear what impact this tropical storm had on the remaining mountain chickens on the island, but it's likely that less than 100 survive. But just prior to this huge tropical storm, there were signs of a nascent recovery or potential nascent recovery of mountain chickens on Dominica. We started to find wild frogs and frogs that were breeding. We found metamorphs, for example, which was really encouraging sign. So actually, maybe there is no longer a need for this facility, but the species is still considered to be extinct in the wild, or functionally extinct in the wild on Montserrat. So this leads me on to the next kind of component of this talk, and that's the next steps for the species. And there is a kind of a nutrition element involved here, and I'll get there in the end. So I mentioned prior to the hurricane on Dominica, there was this evidence for nascent recovery of the mountain chicken frog, which was really, really exciting. And it could be that the reason for this is that the pathogen could have changed over time, or the frogs could have changed or be able to deal with infection and much better. And we also found individuals that were, through our long-term monitoring of the population in Dominica, that were positive for chytrids. And this is a species that are very, very susceptible to chytridiomycosis, that were testing negative on subsequent field monitoring, which was really, really cool and made us quite optimistic. What we do know is that chytrid fungi are they're sensitive to high temperatures and they're sensitive to desiccation. And studies in Australia show that frogs that were considered potentially to be undergoing precipitous decline or even locally extirpated or extinct were actually hanging on in these edge habitats. So rainforest frogs were surviving in habitats that are probably hotter or drier than their typical habitats, so at the edge of the forest. And perhaps there could be something that could be learned from this in how we mitigate against this pathogen in nature. And on Dominica and Montserrat in the Caribbean, we have an ideal model system relative to others to better understand the epidemiology of this pathogen and how to take mitigative action. So yeah, on the island of Montserrat, for example, there's just a handful of amphibian species. The mountain chicken is critically endangered. There's an Eleutherodactylus frog, which is very widely distributed throughout the Caribbean, and then the cane toad, which is invasive. So just 3 species for this simple system, whereas we compare it to trying to understand what might be going on in a more complex system. You know, you've got some, this is probably out of date, but some areas where some nations where you might have many hundreds of species, where it might be quite more of a challenge to understand this pathogen. So I have to say that live food aside, not just live food, but space requirements and many other factors means mountain chicken frogs are in captivity indefinitely is not our vision, and neither is it something that's economically viable. It's really expensive to maintain these frogs relative to other amphibians. And it's really vital that we just don't end up with frogs in glass boxes. For us, it's really important that we try and look at true recovery of the species in nature. So what can we do? Given what I just said earlier, we know that frogs lose control of their osmotic balance when they become sick with amphibian chytridiomycosis. And one of the clinical signs of the disease is they sit in water as a result. And there's, I guess, potential to hijack this response. We know that chytrids die at temperatures above 30 degrees. And if frogs are able to tolerate that, could we use heated water as a mechanism to reduce infection burdens? And this is something we trialed in London Zoo without infected frogs, but just using our group of mountain chickens that are maintained in biosecure facilities. And some of the frogs actually exhibited a preference for this. So we could potentially have these heated baths or heated pools in enclosures on Montserrat, and also do things like reduce canopy cover to make it hotter and drier, increase the amount of rocks in the habitat to again make it hotter and drier through the radiant heat emitted from these rocks at night, and give these frogs a fighting chance. And that's actually what's happening on Montserrat now. So this aerial view you can see in the bottom left, these are semi-wild exclosures that have been established on Montserrat, and into these 27 frogs that were bred in European zoos were translocated to semi-wild enclosures back in 2019. And the vast majority of these frogs are still alive, which is huge for us. And this project is a collaborative project by the Mountain Chicken Recovery Program, of which we're just one of many partners. And again, it's bigger than the mountain chicken, as it's our sincere hope that if successful, these methods could be used to drive the recovery of dozens, if not hundreds of other amphibians whose populations have been devastated by this fungal disease. Now back to the nutrition and diet bit. These frogs are still under human care, and despite it being likely that invertebrates on Montserrat could naturally enter these enclosures because they're not fenced off from above, although there are predator-proof fences around the perimeter of the enclosures, because they're, I guess, semi-captive, semi-wild, we had that duty of care to make sure that these frogs are adequately fed. And given that we know they eat so much, we decided that we needed to establish live food colonies on the island of Montserrat, again trying to use locally collective, maybe not native, live food species to ensure that there's sufficient food source, or in the case of an intervention where maybe if frogs exhibit clinical signs of disease, we might need to remove them from the exclosures and treat them with antifungal baths and kind of get them back to full health before they were returned to the exclosures. And in that case, we would also need these live food colonies to feed frogs if we needed to take more labor-intense husbandry interventions. And we used what we learnt on Dominica to establish these live food colonies on Montserrat. The constraints were exactly the same, but we were able to do this far more quickly given our past experience. I guess the only issue we really experienced here was ants were an issue, and we made sure that the live food colonies, the shelving that they were on, were in bowls of water to prevent the ants getting into the colonies and killing the invertebrates. So it was really nice to be able to take this experience from Dominica and implement it in Montserrat very quickly and very efficiently. This took less than a year to get the viable colonies up and running, whereas previously it had taken us over 3 years. And there's great staff interchange between the two islands and lots of knowledge sharing around how to manage invertebrates, but there was historically when the facility was running on Dominica. And so we do offer the frogs these cultured invertebrates, and especially prior to the breeding season, to make sure that the frogs are able to get into the breeding condition. And what's more is these local live food colonies, these live food colonies on the island of Montserrat, when we feed the frogs, they come out and they're really easy to view and assess their health without too much disturbance, because the species can be quite sensitive to disturbance. So these culturing these invertebrates and feeding them out in the evenings really gives the team an opportunity to assess how the frogs are doing in a scenario where the pathogen, the main driving force of population decline, is still present. So it's really nice that at least in this very small pocket of Montserrat, the sounds of the mountain chicken are echoing on through the night. So these are some references here to kind of that underpin this presentation, which I've never given before. So any feedback would be great. And I have to acknowledge these partners. There's a few more than this as well, but it's a real collaborative effort over many years. And I'll stop there. Thank you for your time.